Hello, it's Arno here from EPCC, one of the course organizers. But before I hand over to Same to actually start, I'll just say a couple of words of introduction about BioExcel. So given everybody who's here, obviously has registered because they found out about the course and they'll have found out, they at least know that it's run as a joint training course between BioExcel and Archer 2, the UK's National HPC Service. So BioExcel is an EU-funded project that aims to facilitate development and uptake and usage of various software packages for high-performance, high-throughput computing that are useful in biomolecular modeling and research. So that includes molecular dynamics package, GROMACS. It includes quantum mechanics package, quantum chemistry package, CP2K, which is of course relevant today, as well as primarily also HADOC, which is a structural docking package. So today's session, as Salome will introduce, relates to preparing to start using doing QMMM simulations that will use CP2K.
Hi everyone, my name is Salome, and I'll be giving this course jointly with Julian Sin, Arno Preme, and Holly Judge. And we posted all the course materials here, which you can access them anytime. And you also have, as Arno said, the course chat where we will add some links in case we need them, and any resource we need. So just to start, I'm gonna give a brief introduction on what we are gonna explain today. We're gonna go through the process to prepare biomolecular systems to run for QMMM simulations in CP2K. There are two ways of setting up systems for CP2K. One is Amber format, and the other one is Charm format. Today we are going to explain the AmberTools package, which is free of use software, and it has all the tools that enable the preparation of biomolecular systems, being a protein, being a nucleic acid, whatever you need. You have all the tools to prepare them. So the first thing I want to make sure is that we wanted to ask two things before starting, is that if you had Archer access, which this has been dealt with by Arno, and the second thing is we ask you to have in the setup, you can see instructions to install an SSH client. You will need an SSH client to run this course. Has everybody one SSH client installed? Or did you have any trouble doing this step? I mean, if you had any trouble, please send some message in the chat, and we will help you to set it up. Okay. Okay, thank you, Pierre. Great. I think that you should ask for that project. Okay. So, while if you got an SSH client and you just requested the access to Y14, that's okay. But you have to request access to Y14 Amber 2020. So I think I will, since nobody else is saying anything, I think we will start. We also recommend that you have some visualization tool installed. We recommend 2 of them, bimol or VMD, but if you have any other tool that you prefer, please feel free to use that. Okay, so let's gonna start. I'm gonna start with a brief overview of AmberTools. AmberTools is, as I said before, is a compendium of several software packages that were developed as part of Amber, which is not free of use, but they distribute this AmberTools suite free of charge, and you basically can use it for whatever biological system of your interest. So it comprises several programs. These are a few. To name, this NAB is Nucleic Acid Builder, which enables you to build molecules and running MD of these systems. Also, you have Antichamber and MCPB, which are programs to create force fields for general organic molecules, and MCPB is for metal centers. Also, you have TLIP and PARM, which are some tools that allow to building Amber topologies, and with PARM you can modify Amber topologies. They also provide this SQM, which is a semi-empirical and EFTB quantum chemistry program that we'll use for very specific reasons. You have Poisson-Boltzmann models, 3D Arism, which also solves integral equations models for solvation. You have Sander, which now is free of charge. Before it was part of the Amber package, and it enables you to run molecular dynamics simulations. We will use this tool too because we are going to run short MD runs. You also have PMD, which enables you to use advanced force fields, and GMX, a program for pushing the boundaries of AmberMD, which is mostly used for parameter fitting. We are not going to use this one today. We're going to use CP2K, which is one tool to analyze and post-process trajectories within AmberTools. And then you have MMPVSA, which is an energetic analysis of MD trajectories, which we are not going to use today. I think that some of these tools can be useful for several people outside for their projects, outside this specific aim of running QMM simulations. So I think that it was worth to look at them, so you're aware of them and you might use it in the future. So Let's gonna start. For those who have Archer access, you should be, we are gonna log in into Archer. Please, if you can run SSH minus capital X, capital Y, user, and login archer.ac.uk and enter your credentials. Do it now. Please let us know if you have any trouble with this. I'm going to give a couple of minutes to do this.
So, Salome, I think that I guess anybody who's using a client that's not simply a command line interface connection, SSH connection, they'll just need to make sure that when they initiate the connection that they tick some box or enable some option that says forward X11, which allows visualization to be taken across from Archer onto and be displayed locally on your machine. So how to do that differs from each client. There are probably instructions. There are probably instructions on the Archer website on for a couple of the clients recommended. Sagar, so I can initiate a chat with you directly, Sagar, to help you with that.
Thank you, Arno. Maybe we can share the Archer. Has everyone else been able to log in? Those who have access? In case you don't have access to, okay, good, Kano. In case you don't have access to Archer, I think the first steps if you have AmberTools installed in your laptop or your computer, you will be able to do the first few steps of AmberTools. The running in the queue, you won't be able to, but maybe you can try that after you get the credentials. Okay.
Just before you get started, Salome, it might be worth just turning off the those pop sound notifications, the audio notifications for pops. It's possible to do if you check the fold-out sidebar and you go to the settings, the gear icon on the right-hand side, actually.
Sorry about that.
Yeah, in there, and then settings. You scroll down, notification settings, and then you could maybe it's just I think the audio in particular. Yeah, it might be useful to turn that off.
Sorry about
Yeah,
that.
yeah. I just thought might be nice to. It's not. It might be distracting to you. So. Okay. So yeah, Julian has just highlighted something that when you request your machine account on Safe, the initial when you request, you just give Y14, and later on, you'll automatically add it to Y14 Amber 2020.
Yes.
Mentioning just explicitly, sorry to butt in again, but just to help people who are applying for accounts avoid the issue that Edna just highlighted. So it is not possible to register in Safe with addresses that are not institutional or organizational addresses. So for example, Gmail or Yahoo or anything like that, Yahoo Mail. So it should be an institutional address, for example, but not necessarily in the UK. It could be .ac.uk or a company or institutional email address. This was in the email mentioned in the email last week as well.
Okay. So okay. I think that you will get the access to Archer granted at some point this afternoon. I hope it's before this session ends. I think I will go through this section, and whenever you have access to Archer, you can go through this part, and then you can follow up the tutorial. Is that okay for everyone?
Yeah, I think if people are encountering issues connecting, they can mention it once in the chat, and then one of us, so myself or Julian or Holly, will possibly message you directly if it's something that looks like it might require a few messages. So sorry, I'll let you go.
No, that's okay. So I just logged in Archer, so the first thing I will have to do is load the environment. So you do this through the tool module. So what we need to do is every software is installed in a module, and you can load the necessary environment for your software and unload as you please. So what we need to do is to type these commands. That I've listed here. So we need to do this model swap at the beginning, so we don't have problems, but then it's just module load, AmberTools /20, then module load propk, and module load open bubble, the version 2.4. Once we have this models loaded, which we can see the module list if we need to be sure we are currently, once you enter in Archer, you are in your home directory. We need to go into our work directory. So we have to do this change of directory to, in my case, it's a different budget, but should not make any difference. So once you are in your work directory, we are going to download some data to run the course. So this data is in a repo, and you just clone the repo here, and it should be okay. I'm going to explain a bit what's in this repository, which is called Files AmberTools. Here you can see that you have several PDB files and several input files, submission scripts, and you also have a folder called outputs. If you're lost at any point of the session, or you have any trouble, in order that you are not lost and you can keep up, you will have here all the outputs, the correct outputs of this session, which are would follow the sections in the schedule. Okay, so if you afterwards get access into Archer, then you can easily pick up using this folder. Is that okay? Okay, so after this, we can start with a proper tutorial. The first thing we are going to do is to prepare our PDB file. PDB file has the coordinates of our system. And in this case, we are going to use the structure of the main protease of the SARS-CoV-2 virus. The structure we are going to use has the identifier 5R84, and if you click this link, you go to the Protein Data Bank webpage of this protein and this crystal, and you see different sections within this PDB webpage. Has a lot of information, is really useful, but has a lot of sometimes it's a bit overwhelming. So the first thing you can see is the identifier, which is the title of this structure. and which organism and where it was expressed, and it has no mutations, which is an important information to take into account. We have the experimental section, this data section, tells us which method was used to obtain this structure, which resolution, which is below 2 angstroms, which seems quite nice, and also the PDB validation, which it shows us in a very visual way, which is the quality of the structure. We can see it's tending to worse, but since you can imagine that this structure was resolved in a bit of haste, it will have to do for this course. So there's a pending publication of this. There's also a view here of the biological assembly of this this protein. You can see here that it's different than the symmetric unit that we have, and that we are going to use this as symmetric unit. So what else is in the PDB webpage? You can find the contents of your PDB. You find under the section macromolecules, you have the protein, which is this 3C-like proteinase, which you only have one chain, and this is the length of the sequence, and 0 mutations. You can see here the protein feature view shows a bit of what's of the biological and the amount of sequences, the biological importance and the amount of sequences that you have and the PDBs that you have for this protein. And in fact, the main protease is one protein that breaks the peptide chain produced by the RNA of the virus and enables that to split that protein chain into different proteins that are important for the virus replication. That's why it's so important. It's an important target that we want to tackle, and that's why this study was used to try to look for fragments or small molecules that bind to it. And in this case, we have that it has 2 small molecules bound. One is the GWS, that it looks properly like a fragment, and the MS, which is a crystallization product. And afterwards, we have experimental data, which is similar to what we have seen before, and how it was obtained, this structure. What we want to model from here is the protein and the GWS ligand. And we want to get rid of the dimethyl sulfoxide, which is not interesting for us. So if we go back to the materials of the course, you will see you have the same information here. So what we have done for this course is to use a short part of this protein to make sure we can run all the steps of the preparation in the time allocated for this course. And also, and we have provided these files here. I'm going to show you what we have here. I'm going to use this is the PDB directly from the PDB, the protein data bank, and this is the structure we are going to use. We are going to only focus on this bit because we only want to see the interactions between the ligand and the protein. So we can get rid of this bit up here. If you want to do this in a proper way, you would consider the whole protein, but for the sake of time, we will keep this short. So the first thing we would do is to split this PDB into different parts. One of it would be the protein, the other part would be the ligand, and the crystallization waters in a third part. So I would suggest to do the grep commands here. So if we look at this file, we have everything here, and we only want to get the protein residues. So we have to get rid of the residues that contain GWS, the residues that contain DMS, and the connectivity, we don't need it now, and the water, crystallization waters. We're also going to do grep of the ligand GWS, like this. And now we will do the same for the water molecules. Now we have split the system into three parts: the protein, the ligand, and the water molecules. So now we can continue. So one important part of setting up your simulation is.
So sorry, Salome, I was just going to, sorry to interrupt. I was just going to suggest that we just take a second to see if that's okay for everyone, and if you know, everyone's managed to do that step.
Sure.
Split things up. Maybe we should do like the hand raising. I think would be useful.
Yes.
So if anybody, which way around? If anybody, the software carpentry approach is usually if somebody is not, everybody starts with their hand up and then lowers it if they have finished. Anyway, anybody who's not, or maybe everybody who has already completed a step, if they can use the raise hand button. Or give a thumbs up, that works too. There's a question, I think, about the viewer. Pymol, I guess, is meant. Vadanavas, did you mean how to call up Pymol or something else?
You won't be able to call it from Archer.
Yeah. Okay.
I just wanted to show what we are doing by grepping, because grep seems a bit you don't know what are you clipping from the file. But I mean, if you can, I provided links to download Pymol or VMD in your laptop, and I think that if you want to do that afterwards, you will have all the files to play with it. Is that okay? Yeah. So, anybody had any problems so far? Okay, I'll take that as a no. But if you have any trouble at some point, please stop me. So I was saying, one important part of setting up your simulation is knowing or trying to mimic the experimental conditions of your biological system. In this case, for this main protease is located in the cytosol of the cell, so we know which pH it would be, and it will know which temperature. Here we are going to use standard conditions for the temperature and standard conditions for the pressure and everything that is standard for running experiments in a lab. And we are going to use the pH of the cell cytosol because that's where the protein is stable. So once we know this, and I want to highlight how important this is, because if you want to compare your results with an experiment, you will need to know which are the experimental conditions to set up your simulation properly. And if it's important that you check the literature of your protein before you start running too. So now that we know what experimental conditions we are gonna do, we want to assess which is the protonation state of this protein at 7.4. It's usually the protein force fields encode the protonation state of the different residues by assigning names to each residue. So a serine is encoded as ser, tyrosine as tyr, but there are some residues that are tritable, so they can change its state because they have a chemical moiety that can change a protonation state depending on the pH. For example, you have an acid like the glutamic acid. So if you have glutamic acid, in the force field you can have blue, which is the deprotonated acid, and GLH, which is the protonated one. So we have some acidic residues like aspartic acid, glutamic acid, and in very extreme conditions, tyrosine, serine, and cysteine can act as acids. And you also have some basic residues like lysine, arginine, and histidine. These are the residues we want to know which state this is in. I mean, since 7.4 is not extreme condition, we can forget about tyrosine, serine, and cysteine right now. There are several ways to check which is the protonation state of a molecule. For example, two well-known softwares to do this step is Propka and H++ server. We have added links to these two software packages. In this workshop, we are going to use Propka. So we have loaded Propka before, if you remember. So if we type Propka31 and the protein file we have just created, you will see that it runs and it gives you an output. All this information is written down in a file called protein.pka, but it also drops all this information through the screen, through the command line. This is the summary of the prediction. You can see that has several tetrable residues, which is this is the residue number and this is the chain. And this is the calculated pKa. So you have to think that at 7.4 you will have 50% of population of one species and 50% of the other. So we have to check which is pKa values that are close to 7.4. The closest we have is this histidine, but it's still 1 pH unit away. So I think we are safe to that we can use this as a deprotonated one. Cysteines are very far away, as you can see. Tyrosines are very far away, as you can see. Lysines are quite far away, and arginines too. So, okay, we have checked. We don't have any residue that we have to change its protonation state, or we have to consider two simulations for each protonation state. That's okay. We're good to go. Oh, I wanted to mention that histidines are tricky residues because they can have 3 protonation states. They can have the hydrogen in the delta position and the hydrogen in the epsilon position. And they can also be protonated with an hydrogen in both. You have to be careful and check which is the best orientation for these hydrogens here. In solution, the most common conformer for a histidine is epsilon. This HIE. But you should always check before trusting and just placing the histidine in this state. In this case, we have some interesting an interesting histidine because, for example, you can see that you have here a histidine that is directly interacting with this with the ligand, and we can see that this is forming a hydrogen bond, and indeed the hydrogen should place here. So once we know this, we have checked the structure, we have checked these histidines, residues, these arginines, everything. We are ready to prepare the protein. AmberTools has a package that can prepare the protein. It's PDB for Amber, -I protein .pdb. First of all, it's PDB for Amber. You have a lot of options here. You can set constant pH simulation, pH simulations. You can mutate. You can do many things with this tool. We are gonna use it just to make sure that the format is right for Amber. So I'm gonna type for Amber -I, which is the input file, and -O protein, which is the output file. So it's gonna output another PDB. If you do this step, you get some information back. PDB for Amber has checked how many chains do you have in your file and how many non-starter residues, how many missing heavy atoms. We have only one chain, that's okay. We also have alternative locations, so we have a residue that has 2 positions in the crystal structure, and the scientists that uploaded this structure in the PDB, what they did is to place both because probably that residue is in 2 positions.
Sorry to interrupt, Salome. There's a question there's 2 questions, in fact. Maybe you already addressed the first question and I may have missed it. So the question from Pierre about, oh yeah, you're seeing it, yeah.
Sorry about that.
I don't know if that's...
I mean, your PDB, your pH influences your simulations a lot. If you don't set the you can have pH trigger conformational change. For example, if you don't set your protonation set right, you, for example, are introducing a charge where there is no charge, so that can create instability of the protein. If you have, for example, if you have a residue that has a value very close to 7 and you want to run a simulation at pH 7, there are, I would say that the correct way to do this is to run two simulations with two protonation states, because you will probably have both in that at that pH. There's another way to overcome this by doing constant pH simulations, which is a complex setup of the system, where you can have your histidine, for example, that can tune between two states, and the simulation will always keep the constant pH value that you feed it. But the setup is not as easy as this one. Is that what you meant, Pierre? No problem. And what if the problem has missing loops? That's a difficult topic, because you have some servers like Rosetta that enable modeling missing loops, and you can also try to find some of this information in another crystal structures that are published from the same protein target that you want to simulate. And if you don't have any crystallographic information from another PDB file, I would suggest you to model it using any of these servers. And hope for the best. Missing loops and crystal structure means that that part of the protein is probably very flexible. So it's going to be difficult to model. Yeah, there are a lot of servers to add missing loops. That's a very good preference. Bim, thank you. I hope I answered, and thank you, Bim, for your comment. Okay. Okay, so going back to PDB for Amber, it gives us the following information. Has chain A, that's the same we saw in the PDB webpage. It points out that we have a residue of two possible orientations, which is alternate, what it means, the alternate locations. Probably when they were resolving the crystal structure, they saw two possible orientation of this residue. And if we have a look to this residue, is this valent here, which is really far away from the ligand we want to study. So I would say that in this case, if what what PDB for Amber does is correct. So it takes the first orientation and deletes the second one. Because since we don't see that this residue is doing contacts with anything else, I think that's a safe approach to do in this case. Otherwise, if this residue was in the binding site, we should be looking very carefully which orientation do we choose. So, okay, I think we have the protein ready. So we have prepared the protein, now we have to prepare the ligand. We have to do the same thing. We have to check which is the protonation state of this residue, which is this small molecule, in these conditions. So there are several ways to do this. There's a very good reference that is PubChem. In PubChem, you have a lot of information, experimental data from a lot of organic molecules. In this case, for our protein, we don't have any experimental data that tells us which is the pKa of this compound. So this is not useful for us. But if you are simulating any of the system, this is a very good reference to look at. So what I do, because it's free to use and it's you have to register, but it's free to use and it has a good visualization setup, is Marvin Sketch. And what you have in Marvin Sketch is you draw your molecule and you calculate the protonation state. It's going to complain about license. And it tells us that at 7.4, the most common state is this one. So once we know which is the state of our ligand, we are gonna protonate it. To do so, there are several ways. You can manually add the hydrogens. You can use bimol to add the hydrogens. Here, today we are going to use OpenBubble, which also is a tool that enables format change of molecules. It's a very powerful tool, which we are going to use a very small part of it. So we are going to type all variable minus IPDB, is the input format, GWS PDB, minus O PDB, GWS with hydrogens, .pdb, I minus H, which is the command that tells OpenBubble to add hydrogens to this molecule. Okay, we got an error. Has not created the molecule, and it's okay. Sorry about this. What it's generating is this file that should be shown here.
Pierre has mentioned something that may be an issue. He said missing the -O.
Oh yes.
I don't know if that's.
Yes, you're right. Thank you for that. So we have now this molecule, and it has created the correct molecule. I suggest you always to visualize it, because in this text format it won't be, it would be difficult to see, but here we have it, and it's the same protonation state we had in Marvin's sketch. So we have protonated the ligand, we have prepared the protein, so we are ready to do the next step. We'll do another section and then we will have a break. Is that okay? Or are we tired? Okay, so the next section is how to parameterize your ligand. Molecular force fields give you parameters residues already parameterized, like all the residues in the protein, residues for nucleic acids, and water molecules. But when we have a small molecule, what do we do? So in this tutorial, we are going to use antechamber and ParmCheck to generate parameters for this small molecule. In this case, the quality of the parameters is not crucial, because what we want to do afterwards is to run a QMMM simulation where the ligand will be treated QM. So we need parameters for this ligand that are good to go to equilibrate all the system before running the QMMM calculation. So these steps we are gonna do here are quite simple, and this is a very simple approach. If you have more complex needs or more complex ligands, this might not suit your needs. And at the end of this page, you will see that there's further information with some tutorials to parameterize ligands. Okay? This is not an easy process, I'm gonna say, but let's gonna do that. Which is the process we're gonna follow. We are gonna calculate point charges using AM1 VCC2 charge method, which is part of which is already integrated within AmberTools, and then we are gonna assign atom types using Amber force field for small molecules, which is called the general Amber force field, which is GAF. Okay, so let's gonna do this. We're gonna use antechamber. Antechamber is a very broad software that if you type antechamber -h, it has many options. You can do many things. You can calculate point charges with another software and feed it to antechamber. You can do, you can ask in antechamber to calculate point charges. You can do many things. So we are going to use the default values, but if you require anything else, I would suggest you to look into the documentation of antechamber, because probably it will you will have an option that will help you. So what does antechamber do? You feed him a structure, a chemical structure, and what it does is understands the chemical structure, identify bonds, identify in and looks for suitable atom types for them, for those bonds, and for that atom description. It also, if you have two atoms that are equivalent, it also detects that, and it, after selecting atom types and generating point charges, writes residue topology files. So what we are going to do is to call antechamber to do this. So we're going to do antechamber minus I is the input file. It's going to be the ligand with the hydrogens. We have to say Fi, which is the format we are using, which is PDB. Minus O, it will be the format of the output, the name of the output, and minus FO, it would be mol2, which is the format of the output. Then we need to tell him which charge method we are going to use. We are going to use the charge method is minus C, which is PCC. The net charge of the molecule is called NC. It was a neutral molecule. And at the end, you can type Rn is the name we want to give to this residue, and which force field he should look for parameters for. And I suggest to use GAF2, which is a more complete graph. So here it's checking the format of PDB. It understands there's no unusual elements. There's no open valence. Checks the geometry, understands the geometry, looks for weird bonds. There's no weird bonds in this molecule, and checks how many units you want to create. And then what it does is use the SQM, which runs a small QM calculation to generate point charges, as we have asked him here. Okay, this takes a wee bit, and that's it. So after this, we need to check. The first thing we need to check is that is the point charges calculation finished correctly. So we have to tail the last line of the SQM out, and you can see that it says calculation complete. So that means it has ended correctly. After this, we can check what we have in the folder. You can see that you have many files that are in capital letters. These are temporary files in case something goes wrong, and you can delete these if you want. You don't need them anymore. And you can see that it has generated this file that we were asking. The Mol2 files are very useful because they contain a lot of information. Sometimes they are not easy to read, but they contain a lot of information that are important. This is the number of atoms, this is the number of bonds, and these are other pieces of information. These are the way that we got the point charges, and these are the information of all the atoms in the molecule. You have the first atom is a nitrogen. This is X, Y, Z. These are the positions. This is the atom type. This is the residue name, and this is the number of the residue that was in the PDB. We don't need this, but it stays here for some reason. And then these are the point charges for each atom. Okay? Here, material, you have more information about the Mol2 format here, but we are not going to go in detail for this. So next thing we're going to do is to check if we have all the parameters for this molecule. And we do that with parmcheck. This tool called parmcheck has all these options, and we are just going to use it to check that generate a file. We are going to generate a file with all the force field parameters for the molecule, GWS. For part 2, you have a very similar working of the software. -I is input, so GWS, the mol2 file that we have created, -F, it's mol2 format, and as we have said here, -S, and we're just going to say F2. I was complaining because I was not giving it an output file. So -O, and we're gonna do GWS FRC not file. Now this file is important to look at. You cannot overlook it. These are all the parameters you need to run this ligand. Here you can see that you have an atom type NS. This will be important later on, and has these bond parameters, these angle parameters, and these dihedral parameters. And also it has found some improper parameters. This looks fine because we don't have any parameter that says I don't have parameters. Param check, if it doesn't have parameters for something, prints 0 value and gives you a comment here that says attention needed. You don't have any parameters on this angle, for example. So please always check this file. Otherwise, you will see your molecule being in a weird way that you don't expect. Okay, so far so good. We have all the parameters for our ligand. The parameters for our protein will be given by the force field. So we are almost ready to build the system. I think it would be good to stop here and take a break. Okay, so let's start with answering the questions that were that rise before the break. Barnabas, we were asking how to choose a force field. That's an interesting question. There are lots and lots of force fields around, and it is not easy to choose one. What I would suggest before you run your simulation is to have a look at some force field. There are a lot of publications that test several force fields against a target for which do they have some experimental values. I would try to look at that publications and trying to see which system, the system that you are trying to simulate, which force field would give you results that are closest to the experiment. Otherwise, you can also check if you're going to use Amber, the Amber force fields are listed here, although you may find some other force field that you can use online. For example, here it points you, Amber web page points you to the Amber 14 SV for proteins, glycan parameters for sugars, and nucleic acids refers to two different ones for the RNA, and I guess that's the DNA one. Also points to the lipid model they have and the force field they have for lipids. There's a reference here which is quite interesting, because sometimes you are using a cofactor, which is there are several cofactors that proteins use, and you need parameters for them. You have the Amber parameter database, which you have several of these cofactors, and you have the parameters and the reference onto which this parameter was derived. You have the same for carbohydrates, lipids, nucleic acids, ions, and different solvent boxes. So this is a good page to have in mind when you are trying to work with a cofactor or something like that. Also, sometimes you have amino acids that have been modified. You have this set of parameters that are compatible with Amber for phosphorylated tyrosines or serines. Also, you have parameters for metal ions, which are not easy to come by. This is another force field for proteins, and this is a force field for proteins that was trained to give values that are similar to the NMR observables. So I would suggest you to look at this. And I'm sure, I know that there are plenty of publications that try different force field against different systems, and that also could point you to could help you to choose which force field you want to use. So the other question we had was from Pierre, and he was asking if we have something equivalent to PROPCA for DNA and RNA. And the only one that I know of is this Delphi pKa, which can give you pKa for DNA and RNA as well. I have never used it, but seems I think that it would be useful for you for your systems. I will post this into the pad. Okay, let's continue with the course. Now that we have parameters for the protein and parameters for the ligand, we're going to continue and build the system, build a file with all the coordinates of our system, and a topology file with all the parameters of our system. To do that, we are going to use lib. Lib is a tool from AmberTools that has two flavors. You can use it through the terminal, which is the command tlib, and you can use it with a GUI that it's called xlib. Today we are going to use the terminal one, but I suggest that if you are learning lib, xlib is quite useful because you can see what you are doing. It has a visualizer. However, I'm gonna warn you, xlib is quite buggy and it can be a bit frustrating, but do not despair. So let's gonna start with tlib. If we type, we enter into tlib and it has a format like a terminal into which we are gonna load, we are gonna write several commands. I am gonna go through it and type the commands myself, but I have added, you can also list all the commands and ask lib to execute them one after the other, and it takes a really short time to do so. So here we need first to build our system. We need to load the parameters, the force field. If we have type list, we have nothing loaded into tlib. So we have to source different force fields. So we're gonna load the force field for proteins. It says that has found the file and has loaded. We're gonna load the force field for the ligand, which is librc-gov2. I'm gonna load also the water model. Tip3P. So we have loaded all these force fields, and now when we type list, we can see that we have a lot of residues that lipids is aware of. Some are like histidines, and some others are leucines, and some others are like ions here. After this, we need to load our coordinates that we have in the PDF file, in the PDF format. For protein, we're gonna load a unit. We're gonna use a unit called protein, which is gonna be the PDB protein for Amber. We are gonna do the same for the ligand, but we are gonna use the mode2 file we have generated. We also will load the Amber parameter file with the command load Amber params, and we are gonna load GWS FRC mode. And last, we're gonna load the crystallographic waters. As you can see, waters in the crystal structure are only the oxygen atoms, so it adds the hydrogen atoms. And it does the same for the protein. It adds missing atoms. Once we have everything, every part of the system we want to build loaded. Let's build the system. Let's call it system, and we are going to combine the ligand, protein, and the vectors. So we'll save a PDB with all this system combined. So save PDB is the command you need to write a PDB, and we're going to write system dry PDB. It's always good practice to look at the intermediate PDB files and the intermediate things you are building with TLib. Since you don't have the user interface, it gives you a better idea of what you're doing. And another useful command of TLib is check. This is going to check if your system is okay. What we can see is that since TLib has added the hydrogens, we have close contacts. Because when it adds hydrogens, it places the hydrogen in an orientation that it doesn't need to be the good one. So I'm going to show you now this. Okay. What we have done with TLib so far is this. We had these three parts, and we have mixed them in this one. You can see the way that TLib adds hydrogens and why the way it adds hydrogens can lead to close contacts between the protein residues and the protein on the bottom. So this means that we will need to minimize this structure and make sure that the water molecules are in a position that makes sense. But this is going to be the next step. So after doing this and assessing that we have built the system we wanted, what we need to do is to solvate the system. So to solvate, you have two commands within TLib. One is solvate oct, and the other one is solvate box. If you want to run a Kuhnemann calculation with CP2K, you can only use solvate box because CP2K doesn't allow truncated octahedral boxes. So we're gonna use solvate box system tip 3p box. We're gonna give this number here is the space you are given for any atom of your simulation to the simulation box end. And then we are also type ISO, which means that we are going to have a cubic box because it will be 10 in every direction. And since our protein is quite round, I think it would be something like that. So after this, it has added almost 13,000 waters. After this, what we need to do is to neutralize the system. Always for simulations, you need to neutralize the system. You cannot have a positive charge or a negative charge in your system. Otherwise, you would see that your system is start to behave in a weird way, and probably your results are not. You can have artifacts for results, and you shouldn't trust what you see. So in order to neutralize the system, you need to add ions. How would you add ions with the add ions command? So you need, say, and a way to know which ions you need to add is check system. The unit is -1. He is sending a warning about this. So what we need to do is to add ions to system and add a positive charge. This step usually requires a bit of time, but it would add 1 sodium ion. We will replace 1 water with 1 sodium ion. After this, what we need to do is to save the parameters, which are in input file. The command is same AmberParam, which is written differently from the load AmberParams. And you have to give your unit, which is system, and system param7 will be the topology file, and a system.rst, which will be the coordinate file. As I said, that takes a bit long. It's also useful to save a PDB file at this point. Let's see if this finishes, otherwise we're going to use the tilip. This is all the commands we want to do with tilip. And what it's doing is generating a system like this, where you have your protein embedded in a cubic box of water molecules. I'm going to kill this and do the same thing, just in a different way. I have written down all the commands I've used here, and I can call tilip like this. If I type tilip -f lipin with, and well, lipin is a file containing all the commands, it would do the same thing. This has finished earlier, and what it does at the end is checks the unit, check everything is okay, and if everything is okay, it starts building the topology. So writes atom parameters, one parameters, angle parameters, all these torsion parameters, and then gives you this summary here. The ligand as your first residue, and you have added 13,000 waters to the system. And now you can see that you have the topology file that looks like this. That has, in this case, we have generated a system that has 42 1,000 atoms, and these are different parameters that it's needed. This file is really long and has information about everything you need to run the simulation. We also save a PDB, and you will see that this PDB contains first the ligand and then starts the protein. And at the end, you have water molecules. It should always also have your sodium ion. Okay, so we have built our system. So I see there are some questions here. TLIP is part of AmberTools, as Julian said, and you have to type source per C and all the force fields within TLIP. And Barnabas has mentioned if it's important to add PDB waters. In my experience, it is because usually crystallographic waters are waters that are very conserved. So there are waters that that don't move and in fact stabilize certain interactions of your system. So probably you have a water that breaches a nitrogen bond with two residues that are and that water is really fixed, so it doesn't move, so appears in the crystal structure. And probably if you are not adding that water, you probably need to equilibrate your system really smoothly and wait for a water to add to enter that position to get a stable protein. In my experience, if you add crystallographic waters, your simulations are more stable. That doesn't mean that you should always check which crystallographic waters you add or you don't add. Because maybe you are adding a new ligand that it's not in the PDB and you don't want any clashes with the crystallographic waters. But maybe you have to delete some of them. Do you have any questions? Yeah, that Barnabas says that step can take a while. If you want to go faster, you can exit TLIP and type TLIP -f lipin, which has all the commands that appears in the boxes, and that will go faster. I mean, you can also wait, and it will get there. Yes, you're right. Elvis has mentioned that there's also another command that it's named add ions at random, and that will make it faster. So now we look here again. We have a system start file and a system parm7. This is the topology, and this is the coordinates. Up to this point, we just prepared the simulation. Now we need to minimize the path in the system, so we avoid any path contacts, and then we equilibrate to bring the system from the state it is in, which is an energy minimization, to the temperature of we want to, and then after that we need to equilibrate the pressure and the density of the box. So we are going to do that now. We're going to start with the energy minimization. I am ask you to submit the simulation to Archer now, and we will go through it because it will take about 20 minutes to complete. So you will have a script that is called send equilibration.pbs, and here you have what an Archer launching script looks like, which has the name of the job. We will send this to the short queue, and we will use 2 nodes, and our wall time is 20 minutes. It will be finished before that. You should have here in the -A flag the name of your project budget. In your case is this one, but in my case it's a different one, so I have to change it. Here we load AmberTools. We need these 2 model commands to do so. We set the working directory, and first we're gonna run a minimization. To do the MM minimization and the MM equilibration steps, we're gonna use the command called Sander. Sander, as I said at the beginning, is runs MD. You can run minimization, you can run normal MD too. So it works in the following way. If you type capital O, it means overwrite any file that has been created before. -I is the input file where we give Sander the parameters to run the simulation, and -O is the output file. The -P is the topology, -C is the coordinates, and -R is the file where we want to save the result into coordinates. For the other steps, the thermalization and the pressure equilibrium, you also see that there is a -X flag. This -X stores the trajectory of the MD. We are going to run the minimization with 8 processors and the thermalization and the pressure equilibration with 48. So, how do we submit a job into Archer? We use the qsub command, and we give this. We give the file we just saw, the send equilibration. It returns us with a number, and if we type qstat, we see all of them. This is my file. This is my job, which is queuing. And but I see all the jobs that are running in Archer right now. If I don't want to start, I should type, and I own -u, that means user, and my username, and I only see mine. We can see that now the job has started running. Here we can see that has started to write the min classical dot out, and it's writing and it's running the session. Okay. Barnabas, you're having trouble with running in the queue because the Archer is you're not member of the budget Y14. Okay, I think Elena that might be the reason, but I would, if you type budget, budgets, you see which project you should be charging your simulation. So maybe here we need to change.
So I think it's possible that for any accounts for which that were only recently created, it's possible that they were not yet added underneath the specific Y14 Amber 2020 budget. That's possible. Okay. Now, let's see. So these are fairly small jobs. They should be able to run under the short queue. I'm just trying to remember if the short queue is charged or not.
It's a very short job.
Yeah, so the short queue is charged, so that means that the small amount of budgets that is currently available within the general Y14 rather than specifically for Y14 Amber 2020, which I think is only 3 cows, isn't it? So that's very little. That won't be enough, I think, to run much at all. So for those people for whom the approval to use Y14 Amber 2020 has not yet been processed due to late sign-up, then okay, so it is currently being chased.
Sorry, Arno, if I can.
I think it's enough.
Yeah, it's definitely enough. Yeah.
Okay. Okay, well in that case, that's good. So if the available budget on Y14 directly is enough, then everybody who has applied for an account, regardless of whether that's already been approved to use Y14 Amber 2020, should be able to run simply within Y14. So in the PBS job script, where Salome mentioned, you should specify dash capital A and then Y14 dash Amber 2020, you should instead just specify Y14. Yeah. Yeah, so these are truly really short, then should be okay. But as soon as people's, I mean, for the next few days, while people still have access to the machine, they should really use the dedicated Y14 Amber 2020 budget, and not the general Y14 budget, which will probably run out fairly quickly anyway. If it does happen to be the case that you're running this practical under an account that is because you're already an Archer user anyway, if you're running in your own with that account existing outside of Y14, then clearly you should use whatever budget code is relevant for the project that that account belongs to.
Okay, should we go through? Or should we wait?
Well, maybe do a quick check to see if some people are maybe trying. So if people have managed to get it to run, I mean, I'll be checking the queue, I guess, and if people have managed to run, can they raise their hand or just let us know in the chat? Sorry. Yeah, no. If it has worked, so if it did work, then please let us know.
I can see
Thanks.
some.
Cool.
Okay. Yeah, I can see some of this running here. Okay. I will go through very briefly through this file here that we used to send. Somewhere has weird uses weird names for things, but I means equal 1 means perform animation, and we are giving two other commands. Max cycle is the number of maximum cycles of the whole minimization, and n cycles is the number of cycles that we'll do in steepest descent before changing to conjugate gradient. We are using two different algorithms to minimize, two different methods, because it's less probable that you get stuck in a local minimum. So my minimization has finished, and what I can see here is that has the details of the minimization. Here is the input file, some other data. These are the flags, all the flags in Sander. It starts with the first we have a very high energy and a very high Gmax is the slope of the potential energy surface of the system, and this is quite high. Ideally we want the lowest energy possible and the lowest Gmax possible. And if we go at the end, the final results are that the energy has decreased quite a bit, and the Gmax is 9, which is could be better. It would be the closest to 0 would be the best, but it's not too bad. It also tells us the atom name and the atom number that it's causing this Gmax value. So we can, if the minimization crashes, we can know which atom is causing it at each step of the minimization. So that was the minimization, the energy minimization. Do you have any questions regarding this section? Okay. People doesn't have questions. So we're gonna go through the thermalization and density equilibration. We have minimized our system, which means that we have only minimized the potential energy. We haven't added any part of the kinetic energy that would be linked to the temperature. So we need to bring the system smoothly from 0 Kelvin to the temperature, to the ideal temperature we want. Our target temperature is 298 kelvins, which is 25 degrees, which is the standard value for room temperature experiments. So the first part we're gonna do, we're gonna raise the temperature in an NBT ensemble, which keeps the volume constant. And afterwards, we're gonna do a pressure and a density equilibration, which is gonna be M. And we are gonna allow the size of the box, which would be the volume, to decrease as much as is needed to fix the density of the box. In the thermalization, we can see that this is the input file. If we run imin equals 0, it runs molecular dynamics. It doesn't minimize. This NTX, it tells Sander to read the coordinates, but not velocity, if we choose the option 1. If we use irest 0, is we are generating velocities for this system. This is the number of steps we are going to perform. This is the time step. This is the constraints of the hydrogen bonds. And we are going to go from 0 to 290. And we're going to save, I'm going to print the output every 500 steps. This is the cutoff for the non-bonding interactions. Constant volume is MTB equal 1, MPT equals 0, it means no pressure scaling. And for the temperature, we're going to use the ligand thermostat, and these are the parameters. It is important to notice this flag here. This is the random number generator for the velocities. If you type -1, it will get a random number. If you type any number you like, you could choose whatever number you like. That would be the seed for the random number generator. So if you want reproducibility, you should check this IG option. Then we have the option NMR opt, which enables us to give Sander more parameters, and we are choosing to create a temperature run. We will go from zero here to 298 in the first 12,000 steps, and it will be constant the last 3,000 steps. and this will give us these three files: the output file, the resulting coordinates and velocities, and the trajectory file. You should see something like this in your output file, but since we are randomly generating velocities, it will change. It prints this format for its 500 steps, and we can see that we have reached the temperature we wanted and have no pressure because we are not using this the MPT ensemble, and it prints several other bits of information that are going to be important. For the pressure equilibration, we have a similar output. We have the same length of simulation. We are telling to read both coordinates and velocities here. The same thing about printing. The only thing that changes is MBT. Now is constant pressure, and MPT1, which is the pressure scale. It will also give you an output, which is the log file of the simulation, the resulting coordinates, and the trajectory. And here you will see that the equal classical out file has the similar format, number of steps, time, temperature, which is close to what we want, and the pressure. Pressure values are quite crazy, but what we need to check is the density. The density should be close to 1. In this case, it's quite close, but it doesn't seem to have converged. It's close to converging. I want to highlight to mention that all the runs performing this course are really short, and if you have you're simulating a system of interest for you, you should consider seriously expanding to this to some nanosecond scale. So here it's important that we analyze the equilibration simulation. We have seen that we had the number of steps, the time, the pressure, the temperature, the pressure, the total energy of the system, the kinetic energy of the system, and the potential energy of the system. What Amber provides is a Perl script that gathers all this information and prints a really to use format in any of the plotting software you want. It's process MD out, and you just attach the two outputs we have seen. We're gonna start with heat classical and then equilibrate the classical, and it prints a lot of files. You can see, for example, the ready to use format I was mentioning. It starts at 0 steps and goes through it till the end. So if we check our simulation works that we choose to print to plot this in a nice way, we should see something like this. Our system was heated smoothly, and then it reached a baseline. which also is translated in the total energy and the kinetic energy. You should see that these values tend to converge by the end of the equilibration. If they have not reached a plateau, you should expand your equilibration step. When we look at the pressure and density, we see the same thing. The pressure should be at 1, but we can see that the fluctuations of the pressure are quite crazy, but they are around 1. And the density of the system is not considered through the thermalization simulation, and it's equilibrating in the part, in the equilibration, density equilibrations part. And these two should reach a plateau. If you do this analysis and you haven't reached that, you should consider extending your equilibration simulation. You can run many more analyses of this equilibration step, but this is not the purpose of this course. But we are providing a Jupyter notebook with the code to create these files if you want. It's in the GitHub repo you downloaded. So we are almost ready to submit a QMM calculation. We have to fix some bits. We need to add some missing Lennard-Jones parameters of the force field, because otherwise you would have a weird behavior between the QM region of the QMM calculation and the MM region, and that can lead to some artifacts. To do so, we are going to use PARM. I'm going to stop and ask if you have questions from the previous section before continuing this. Is that everybody following? I'll take this, yes. So we are gonna use PARM. PARM, as we said at the beginning, is a tool that enables us to modify Amber topologies. And as I said, there are some missing Lennard-Jones parameters in the force field, and they are particularly the hydrogen atoms. Some of the hydrogen atoms doesn't have Lennard-Jones parameters, so we have to fix that. So PARM works in a similar way as has a cool prompt, and you have to load your topology file, and for example, we can identify which are the atom types need to fix by using the print details command. For example, this is a water molecule, and this is the hydrogen of a water molecule. It has no Lennard-Jones radius and no Lennard-Jones depth parameters. So we have to amend this. In order to do so, we use the change Lennard-Jones single type command, and we have to fit a single type here, and we are going to add these values here that they are the values of the hydrogen atom of the hydroxyl group in GAF2. I chose these parameters from there. If we type this, it mentions changing for this atom type, this value to this value, and this value to this value. If we do the same with the other hydrogen atom of the water, since it's the same atom type, it's already the same thing. I suggest you that you change also the serine hydrogen atom and the tyrosine hydrogen atom. Once we have changed this, and you have here the same behavior, this atom type was the same as this atom type. How can we see which parameters are missing? So far, PARM gives you a lot of options to play with, but the one I have found more useful is sprint details, because you actually fit the atom number, and that gives you the details of that atom number. And it's usually all the hydrogens in the hydroxyl groups and this kind of moieties. Also, you can check that through CP2K has an option that tells you have missing parameters, please check this. That's a good question, Kono. I haven't found a way, but this is the best of my knowledge. Sorry about that. So at the end, we need to save again another topology file. So we use the command outparm and writes an output file. And then we just type quit. If we type ls, we should see that it has written this file here. Another thing we need to do is to pre-center the solids in the simulation box, just in case. With the short simulation we have done, it wouldn't be necessary. But I think it's a good practice before starting a production run to pre-center everything, because it will make your life so much easier by the post-processing trajectories afterwards. To do that, we are going to use very briefly the CP2K tool. CP2K works in a very similar way of PARM. And this is we load the topology. And we just have to load the input trajectory, which was system LST7. Now I'm going to use a command that is really useful, that it's AutoImage, that usually fixes all the problems with the PVC of your simulation. If you have a complex, it might not do the job, but if you have single chain, a single protein, which is a single chain, it fixes the problem, and it centers the protein to the center of the box. And right now, we just have to track out which is output a trajectory file. That is, and we're gonna name system equal 0 RST7, and now we can output any format we want, almost every format we want, but we need to specify at the end which format we will want. So we have listed all these commands. The difference between Parm and CPP trash is that Parm was doing all the commands on the fly, and here you have to list all commands and type go or run, and then it executes all the commands. And afterwards we can quit, and that's it. Here we have the system jump mode Parm 7 and the system equilibration 0 RST, which has the correct format and the protein center. Now we are ready to run the QMM calculation with CP2K. You have any questions regarding this? I mean, CPP trash can do many things because it's a tool that can analyze trajectories. It can do very complex analysis, but we have just used a very simple thing to do with it. Everybody following?
So I'd like to suggest that I think for the given the time we have until the hour, and I think the actual video call session I think is due to end actually at 4 o'clock. So we just need to watch out that we don't all get kicked out at that end time. But even though there might not be enough time to go through all of the actual running of the CP2K jobs. And anyway, the main purpose of this practical was, of course, to prepare the systems to run with CP2K for QMM. Maybe it makes sense to highlight so Julian just commented that actually the session might not be terminated exactly at 4 o'clock. So it might be true, but it might be worth highlighting some of the steps involved in running with CP2K, and maybe in particular some of the things to take care of. And I don't know whether you or Holly wants to do that either. Actually, I'll take just 1 minute at the end to say something quick about feedback and about further QMM support from BioXL, if I may.
I mean, I will try to be short. For the QMM run, what I would suggest you as a next step is to run a QMM monetization run, which means that you are not running the production yet. You just are checking that the QMM run works fine. So it's just another equilibration step, making sure that your QM region behaves as expected and your MM and QMM interact in the way you want. So we have added as a last bit how to run this QMM monetization. Here I give a brief introduction to CP2K. CP2K is a quantum chemistry software tool that it can perform many simulations of many flavors. You can has a lot of modeling methods. It has DFT, semi-empiricals, almost whatever you need, and it's also compatible with Amber and Charm force field and GROMACS. And you can do normal MD, plain MD. You can do sampling dynamics. You can do vibrational analysis or other spectroscopy analysis. You can. It's really powerful. So I suggest you have a look here and look into the documentation here. What I am gonna explain now is that we are gonna prepare a QMMM monetization run that will use only the ligand as a QM region, and it will use a semi-empirical method PM3 to define the QM region. CP2K uses an additive QMMM scheme, which means that the QM region will be treated apart from the MM, and then the interactions from the MM part and the QM part will be computed per se and added to the energies of the Hamiltonian of the system. So what I have here is send QMM calibration, and we have this file that's again sends a very short simulation using the latest version of CP2K, and CP2K here just needs the input file and can output a direction to the output file to the log. The CP2K input file is really long, and it gives you can tune many bits, but that makes the input file really long. I have provided a commented version here. If you go to extras, you will find CP2K input file. Has explained every section of the CP2K input file, so you can have a look at any part that you have questions. I just will point you to the parts that you need to fix. First, this is a CP2K input file. You have a global section that basically states how the files are named and which type of simulation you're running. And then you have the force evaluation section, which is where you define all the QMMM sections. The method you're going to send is QMMM. Here in the FT, you specify the parameters for the QM part. You also have the MM part, which you have to specify the topology of MM part. And in the subsystem section, specify how big is your system and which is the topology again and which are the coordinates. Here you can specify also any other atom type that is not normally defined as part of this system. Here in the QMMM, you specify the details of your QMMM simulation, as well as the atom indexes that are required of the QM region. For example, we have QM kind, nitrogen, or atom indexes of 1 and 10. And then we have the section motion that we tell to run a short MD run. We are running just 1,000 steps with a short time step. We keep the temperature, we set a biostat, and we do the same thing. We set a thermostat, and we give all the printing information you need. So here you, as I said, you need to add the Amber topology and the coordinate files in this subsection and the QMM force field section. You have to add the box size of your system in the cell part of the subsystem. This is really easy to do. You just need to tail this last line of the this precentered trajectory file. You have X, Y, Z, and these are the shape. This is the shape of your box. And as I said, in the QMM section, you have to add the atom indices. It's very useful to place the ligand, the first residue, in your simulation, so you can easily find the ligand and the atom indexes. And I think the CP2K output is will generate these files, which is the weight function, the shape of the cell, the energy, the trajectory, and a restart file for the simulation, and a log of your simulation. And at each step of your QMM run, you will find this kind of output. It has this shape, the number of electrons, the occupied orbitals, prints all this all this information, has a section for wave function optimization, and then it prints the energy of each step and the variables of your system at each step, meaning temperature and pressure and volume. I think that the system, you can run it like this, but we won't have time to analyze it here.
So I just noticed mention that so there's a couple, I see people have asked some questions in the chat about while doing QMM with CP2K. Perhaps you can already say something, Salome, about this, but even if not, and this is something I was going to mention basically at the end of the session as well, is that if anybody has any questions, either directly relating to the practical, or indeed actually more widely doing biomolecular simulation with CP2K using QMM, then BioXL has a forum, and this is linked to from in the scratch pad for the session. We see the link to it says for further assistance after the course, go to ask.bioxl.eu, and there's a forum there where you can ask questions. So even if we may not be able to address all questions right now in detail, I would really encourage people to ask these again, or to modify them and ask them on the forum after. But if you want to provide any comments now, Salome already, or Holly, if regarding CP2K, feel free.
I mean, if Holly, feel free to jump in if you want. Stefano is asking if is the modification of hydrogen Lennard-Jones parameters that are equal to 0 only necessary when you run QMM? For CP2K, it is necessary. I am not sure about Gaussian and Orca, but I think it makes probably it also it's necessary for them, because What you don't want is, since you don't have Lennarton's parameters, if you have any of these hydrogens in your QM boundary, you will have you will have weird interactions between the QM and the MM, and that would make your simulation blow up. So I guess that it should be fixed in Gaussian and Orca too. And it's not just a good practice, if that's what you meant. I think that Amber's built-in semi-empirical QMMM takes care of these things, but I'm not 100% sure. Bean was asking, how can we know the cell value in the QMMM setting will affect the calculation? You need to modify manually the size of the cell because it's not able to read it from the input file, from the coordinate file. So you don't want to have a box that is too big, because you can run with PBC, with periodic boundary conditions. So you need to have a box that is quite the size you want. Otherwise, you have weird things happening at the boundary of your cell box. If you have to also set up a cell section for your QMM, for your QM region, you should, I mean, you can make it bigger than actually your QM region, but I don't think you can make it smaller than the QM region you have. So I would look at what is my QM region and add a number that is a bit bigger than what it should be. The other question you had is, I want to include some residues of the QM region. What should I do to separate the QM and the MM regions? That's a very good question, because, for example, if you wanted to use this system to estimate the free energy of the binding, and you have a residue that is directly interacting with your QM region and you want to add it to the QM region, you can do the same thing we were doing. You have to add the atom indexes here, and then you should add a section for the link atoms, because probably if you add a residue or part of a residue, you will split a covalent bond and you have to add a link action to deal with that interaction, that bond that it's in between the MM and the QMMM. And your last question was, how can I read velocity from previous QMMM equilibrium for predictive? Holly has just posted the link file, the link atoms documentation for CP2K. How can I read velocity from the previous QMM equilibrium for productive? You mean, I understand that you mean that you want to read the velocities of the Amber format in CP2K instead of assigning new velocities in CP2K. I think maybe Holly, you can you have an answer for this. I'm not sure. How can you read the velocities? I'm sure.
So, sorry. So I know that CP2K can read in velocities in like an XYZ format through the coord section.
Holly, you
So
sound a bit robotic.
Oh.
But you can add this section in the CP2K input file if needed. That's what I understand.
So just in the interest of time, and because I know that even if the session doesn't get terminated, we still have, I mean, people may have commitments and things like that. So I'll try to, are there any more things that you want to mention to round off the what we were describing about QMM before that, Salome?
I think I'm done here.
Okay, great. Thank you very much. Okay, I will give. So thanks very much, Salome. I will give a quick just take over and share the content for everybody before people have to leave. Let's see. Okay, so people should see the course scratch pad that I'm sharing, although it's far too small. So thanks everyone very much for attending. I hope this was useful. So as I said before earlier already, you will continue to have access to Archer for the next few days until the end of the week. So I would definitely encourage you to finish off running any preparatory steps that you've not yet managed, and also try and run CP2K as the instructions show. And that's stop my. In the Scratchpad for the course, you see there's a link to the BioExcel forum, to the section on the forum that relates to QMMM. So as you see, there are not many queries there right now. So I would urge you, if you have any questions about when you're running through the practical yourself, please ask them here. I think you need to sign up to create an account. Hopefully that's not too much trouble. And also in future, if you have any other questions, even if they're not related to this practical, but with regards to using CP2K to do QMM simulation of biomolecules. So BioExcel is looking to support this. In fact, BioExcel is also developing an interface that allows GROMACS to be used in combination with CP2K, with GROMACS handling the MM side and CP2K handling the QM side. And this is due to be released later this year to become publicly available, and that functionality will also be supported via this forum. So please do keep an eye on that. Then finally, there's also the there's feedback. So we would like to ask that after the course you provide feedback. Or actually, this is on the page for the session itself, which I can share. So at the bottom of the page, you will probably get an email about this as well, asking you to complete this. There's a link to feedback, and you don't necessarily, not all of it is necessarily relevant. For example, accessibility of a venue is up to yourself in these socially distanced times. So hopefully your venue where you attended this course was accessible. But for any of the fields that are relevant, we'd be very happy if you could provide comments. It's very useful for us to know, both for Archer servers, and also or I should say Archer 2, and also for BioExcel to know what was useful about this, about how we can better support your needs, maybe other training that you're interested in with regards on this topic or related topics. We already have some plans to provide further training, but we'd welcome your input on that. So I think those are the main things that I want to mention before finishing. Yes. Okay. So thanks again, everyone, and do get in touch on the forum, or you can email my email address in the chat. Feel free to email me directly, and we can be in touch about this. So thanks everyone, and have a very good rest of your afternoon or evening, depending, or morning, depending on your time zone. Okay. Goodbye, everyone.